DNA replication shows up everywhere on the MCAT, but can you quickly walk through it under pressure without mixing up the enzymes or steps? In this video, we're stripping it down to the high-yield essentials: what happens at the replication fork, who the key players are, like helicase, primase, and DNA polymerase, and how leading and lagging strands actually work. By the end, you'll have a clear, test-ready framework you can recall in seconds. DNA replication is a semi-conservative process in which each new DNA molecule contains one original strand and one newly synthesized strand. This means that when DNA replicates, the two strands of the parent double helix separate, and each serves as a template for building a complementary strand. The result is two identical DNA molecules, each composed of one old strand and one new strand. This concept was confirmed by the famous Mendelssohn-Stahl experiment using nitrogen isotopes, which is a classic MCAT favorite. They grew bacteria in heavy nitrogen and then switched them to light nitrogen, demonstrating that DNA replication produced hybrid molecules after one generation. A helpful way to remember semi-conservative is semi equals half, meaning half of the original molecule is conserved in each daughter molecule. This is important because it ensures genetic continuity while still allowing replication. On the MCAT, you may be asked to interpret experimental data or predict outcomes if replication were conservative or dispersive instead. Also, this concept connects to mutation propagation, because errors introduced during replication will persist in one of the daughter strands. Understanding semi-conservative replication is foundational for grasping how genetic information is faithfully transmitted from cell to cell. DNA replication begins at origins of replication and proceeds bidirectionally with the formation of replication forks. An origin of replication is a specific DNA sequence where the replication machinery assembles and the double helix begins to unwind. In prokaryotes, there is typically a single origin, while eukaryotic chromosomes contain multiple origins to speed up replication due to their larger size. Once replication begins, two replication forks form and move in opposite directions, creating a replication bubble. Each fork is a Y-shaped structure where DNA is actively being unwound and copied. Helicase is the enzyme responsible for breaking hydrogen bonds between base pairs, allowing the strands to separate. Single-strand binding proteins stabilize the unwound DNA, preventing it from reannealing, while topoisomerase relieves torsional strain ahead of the fork. A useful mnemonic is helicase unzips, SSBs stabilize, topoisomerase untwists. On the MCAT, you may need to identify these enzymes and their roles, or predict what happens if one is inhibited, such as increased supercoiling if topoisomerase fails. This concept also connects to DNA damage and repair, since stalled replication forks can lead to genomic instability. DNA polymerase synthesizes new DNA strands in the 5-3 direction by adding nucleotides complementary to the template strand. This directionality is crucial because DNA polymerase can only add nucleotides to the 3-hydroxyl group of a growing strand. As a result, the template strand is read in the 3-5 direction. A key implication of this is that DNA polymerase cannot start synthesis from scratch. It requires a short RNA primer, which is synthesized by primase. The enzyme then extends this primer by forming phosphodiester bonds between nucleotides. DNA polymerase also has proofreading ability via its 3-5 exonuclease activity, which allows it to remove incorrectly paired nucleotides and replace them, increasing fidelity. A common MCAT trap is confusing synthesis direction with template reading direction, so remember, build 5-3, read 3-5. This concept is often tested in questions involving mutations, enzyme deficiencies, or experimental setups where directionality matters. It also connects to transcription, where RNA polymerase follows similar directional rules. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously as Okazaki fragments. Because DNA strands are anti-parallel and DNA polymerase can only synthesize in the 5-3 direction, replication occurs differently on the two strands. The leading strand is oriented in such a way that DNA polymerase can follow the replication fork and synthesize continuously. In contrast, the lagging strand runs in the opposite direction, so it must be synthesized in short segments called Okazaki fragments. Each fragment begins with an RNA primer and is later extended by DNA polymerase. These fragments are then joined together by DNA ligase, which forms phosphodiester bonds to create a continuous strand. A helpful mnemonic is leading equals continuous, lagging equals lagging behind in pieces. On the MCAT, you may be asked to identify which strand is which based on directionality, or predict the effects of inhibiting ligase, which would prevent fragment joining. This concept also ties into replication speed and efficiency, as well as mechanisms of DNA repair. Key enzymes such as helicase, primase, DNA polymerase, ligase, and topoisomerase coordinate to ensure accurate and efficient DNA replication. Each enzyme plays a distinct and essential role in the replication process. Helicase unwinds the DNA double helix by breaking hydrogen bonds, creating single-stranded templates. Primase synthesizes short RNA primers that provide a starting point for DNA polymerase. DNA polymerase then extends these primers, adding nucleotides in the 5-3 direction and proofreading for errors. DNA ligase seals nicks between Okazaki fragments on the lagging strand, ensuring strand continuity. Topoisomerase relieves supercoiling tension ahead of the replication fork by cutting and rejoining DNA strands. A useful way to remember the sequence is unwind, prime, build, seal, relax. On the MCAT, enzyme function questions are extremely common, especially those involving inhibitors or mutations. For example, if ligase is defective, you would expect fragmented lagging strands, while a helicase issue would halt replication entirely. This concept also connects to pharmacology, as many antibiotics and chemotherapy agents target these enzymes to disrupt DNA replication in rapidly dividing cells.